Sjögren's syndrome is named after Dr. Henrik Sjögren, who first identified this autoimmune disease in 1933. Most patients are women, and onset is usually at age 40 to 60 years. In Sjögren's syndrome, the cells of the immune system attack the body's exocrine glands, namely the salivary glands and the lacrimal, or tear glands, resulting in the two most common symptoms, dry eyes and dry mouth. In a normal and healthy immune system, cells of the immune system seek out and destroy foreign pathogens that could harm the body. Macrophages and dendritic cells are also referred to as antigen-presenting cells, and once they recognize pathogens, they ingest them by extending themselves around the pathogen and engulfing it. In combination with major histocompatibility complex class II molecules, MHC class II molecules, the antigen-presenting cells bind peptide fragments derived from the engulfed pathogens and display them on their cell surface. The antigen-presenting cell then searches for a T cell that recognizes the specific displayed antigen. Once the antigen-presenting cell has found the appropriate T cell, binding occurs, which activates the T cell. Once activated, T cells secrete pro-inflammatory cytokines, which magnify the immune response by recruiting more immune system cells, ultimately leading to inflammation. While the exact cause of Sjögren's syndrome is unknown, it is thought to be a combination of genetic and environmental factors. Genetic factors may include genes which code for specific types of MHC class II molecules, called human leukocyte antigen, or HLA genes. The genes thought to be involved are HLA-DRW52, DQA1, and DQB1. Environmental factors include an infection of the exocrine glands, namely the salivary and lacrimal glands. Infections can damage the cells of these glands, resulting in exposure of the inner cell contents and nuclear components, such as DNA, RNA, ribonucleoproteins, and histones. In individuals with Sjögren's syndrome, T cells get inappropriately activated by these nuclear components, which they mistake as foreign when in fact they are self. The activated T cells in turn activate B cells, which start producing anti-nuclear antibodies, or ANAs, which are antibodies against these nuclear antigens. In Sjögren's syndrome, ANAs are formed against the ribonucleoproteins SSA and SSB, and they are known as anti-SSA, also called Rho, and anti-SSB, also called La. T cells and the ANAs are transported via the circulatory system to the exocrine glands in the body, where the T cells secrete cytokines, triggering an inflammatory cascade, attracting more immune cells, leading to inflammation of the exocrine gland tissue. The cytokines also activate fibroblasts in the tissue of the exocrine gland, which then produce fibrous tissue, replacing the damaged gland tissue. The end result is a loss of secretory cells in the glands, hence reduction in the production of lubricating mucus in the eyes and mouth.